Hello everyone, welcome to today's video. Today we will be having a discussion on anatomy of dicot leaf. In my previous session, it was about the topic anatomy of monocot leaf. I hope that it is clear to you now. Based on the posture, there are two kinds of leaves. They are dorsiventral or bifacial leaves and isobilateral or equifacial leaves. Most of the dicot leaves are dorsiventral, but most of the monocot leaves are isobilateral or equifacial leaves. In last session, we have discussed about the isobilateral or monocot leaves. In this session, we will be discussing the anatomy of this dorsiventral or dicot leaf. A cross section of dicot leaf shows three different regions. They are epidermis, mesophyll and vascular region. This is the picture showing the different regions of a typical dicot leaf: epidermis, mesophyll region and vascular tissue. Let's learn one by one. First region is the epidermis. As you all know, it is the outermost protective layer. This pink color region is the epidermis. It is the outermost protective layer present on the upper and lower surfaces of a leaf. And it is made up of single layer of closely packed parenchyma cells, and which is uniseriate. And a thick cuticle is present on the outer wall of epidermis, and this cuticle which checks excessive evaporation of water from the surface, and it provides protection against pathogens and also protects the internal tissues, these internal tissues, from mechanical injury. And another character is that numerous small openings called stomata are present in the epidermis. Okay, and these stomata are especially for transpiration and gaseous exchange. This opening is called stomata. And these stomata are more abundant in the lower epidermis of a dorsiventral leaf. Second region is the mesophyll. It is the thin-walled chlorangymatous green tissue seen in between the upper and lower epidermis. So this green colored tissue is the mesophyll region. In dicot plants, in dicot leaves, mesophyll has two different parts formed of two different types of cells. They are the upper palisade mesophyll, formed of this kind of palisade cells, and lower spongy mesophyll, formed of spongy cells. And these palisade cells are closely packed and vertically elongated chlorangymatous cells with numerous number of chloroplasts and little or no intercellular spaces. And these palisade cells are the major photosynthetic centers of the leaf because they are close to the upper epidermis or close to the upper surface, so that they can receive maximum sunlight for photosynthesis. And the compactness of the palisade parenchyma depend upon the intensity of the sunlight to which the leaf is exposed. And the leaves which receive high intensity of sunlight will have more compact chlorangyma than those which receive low intensity light. So these palisade cells are especially for the photosynthetic function, with the numerous chlorangyma are present in the palisade cells. Here you can see that the palisade mesophyll cells next to the upper epidermis, and they are closely packed and vertically elongated cells with numerous chlorenchyma. Okay. Here you can see the spongy mesophyll cells. The spongy cells are present just below the palisade and extends up to the lower epidermis. This is the palisade cells and this is the spongy cells, and this extends up to the lower epidermis. And the spongy cells are thin-walled and irregularly shaped and loosely arranged chlorenchyma cells with the large intercellular spaces. But in the case of palisade cells, the intercellular space is little or no intercellular space. And spongy cells are for photosynthesis and gaseous exchange. So in mesophyll, it is the region between upper and lower epidermis, and it is differentiated into palisade cells and spongy cells. Palisade cells are located on the upper surface, and it is a closely packed vertically elongated chlorenchymatous cells, especially for photosynthesis. And below that the spongy cells, which extends up to the lower epidermis. They are thin-walled, irregularly shaped and loosely arranged chlorenchymatous cells with large intercellular spaces, and these cells are for photosynthesis and gaseous exchange. Next important region is the vascular region. Here the metric veins and veinlets of the leaf form the vascular system, and here many vascular bundles are distributed in the spongy mesophyll cells, and these vascular bundles are conjoined, collateral and closed. Conjoined, collateral, closed means conjoined means they are the xylem and phloem lie side by side, and in between xylem and phloem there is no cambium, so the bundles are closed. And each vascular bundle is surrounded by a parenchyma sheath called a bundle sheath, and that bundle sheath is extended towards the upper and lower epidermis for their mechanical support, and that is called bundle sheath extensions. So in short, we can see that a typical dicot leaf contains three different parts: upper epidermis and lower epidermis, mesophyll cells, which is differentiated into upper palisade cells and lower spongy cells, and in between that we can see that vascular bundles, conjoined, collateral and closed vascular bundles. These are all about dicot leaf. Thank you for watching.